Actually, yeah, exactly.
At least I don't have to stand behind the computer. Yeah. Yeah, this is weird. It's like I've never done this before. Hey.
There's like
Wait a minute. If you're the only person on Zoom, you have to share your screen, right? Because Zoom won't see this. Yes. Yeah. Yeah. Well, I've never been known for constant. Yeah, we got the I gotta do. We're going to get started right on time in about 1 to 2 minutes. Our speaker needs to get to the Jet Express by 8 o'clock. I'm going to just look around the room. You know what? I'll start with a few announcements. So welcome to Stone Lab's weekly lecture series. We do this every Thursday night. So those of you who are staying for 4 to 8 weeks, I'll see you every Thursday. How many people are staying for 4 weeks? Okay. I know there has to be more than one because there's got to be some more. I know Zach's class has at least 4 students. So I should have seen at least 4 hands. How many people are staying for at least 4 weeks? So how about at least 4 weeks? Okay, sorry, good. There's a few again. Okay. All right. So yeah, every Thursday we'll be in here. We'll also have these broadcasts on Zoom. We thank you to everyone who's online. At the end, what's it? We just made it like last week, okay. Or not last week, earlier today. So there was an email sent out by Jill Gentes, our communications director. If you're subscribed to Ohio Sea Grant or Stone Lab newsletter, you'll get it. We're going to make the rest of them more social media friendly. In 8 weeks, so our last ones, the guest speaker will be our REUs. So we have 4 REU students, research experience for undergrads. They will tell us about their research. My last note on here is just remind everyone that if you're leaving, if you're here for 1 week, checkout is Sunday. All right. So our guest speaker tonight is Dr. Steve McMurray. He comes all the way from Huron, Ohio, right down the road at Olin Creek. And rather than me introduce our speakers, I'm going to let our speakers introduce themselves, and they'll tell us like how they ended up in their current role. And he's got again, we're keeping this one a little bit shorter, where you're either your pleasure or dismay, but our speaker's got to be on the 8 o'clock Jet Express. So with that, let's thank Dr. Steve McMurray. Tell us about what's happening at Olin Creek.
All right, thank you. So as you guys are all pondering, or perhaps pondering your career trajectory, not too long ago, or maybe long ago, I was kind of in your shoes. So I went to Hiram College. I'm originally from Northeast Ohio. And it was really these type of research experiences, either classes at a field station. I had 2 internships, one at the University of Maine, one at the University of Southeast Alaska, but that's really what got me interested in biology. I was also interested in chemistry, didn't know how to combine those 2 fields, but I found a field called chemical ecology. and that's what led me to study marine sponges on coral reefs. That's where I got my master's at the University of North Carolina, Wilmington. I was really, after my master's, interested in how we can use science to inform management and policy. I was lucky enough to be selected as a NOAA Sea Grant Knaus Fellow and served a year in Washington, D.C. in the National Sea Grant Office. I then went back to the University of North Carolina, Wilmington for my PhD. So for my master's, the main finding was that sponges are increasing on coral reefs while corals are dying. So I was interested in the so what. So sponges are increasing. How may that affect the ecology of coral reefs? We know that sponges pump a lot of water. They filter things out of the water column. They're important in nutrient cycling. So these were the chapters of my PhD. How much water do they pump? How much do they feed? How are their populations changing? And therefore, how are they influencing the flow of carbon on coral reefs? With my PhD, I figured I might go in academia, so I probably needed some teaching experience. So I spent a year in Pennsylvania as a visiting assistant professor teaching. Was lucky enough to get a grant to study what's known as the sponge loop hypothesis. So this was published in Science at the time. European authors, they said that sponges were unique in that they could consume dissolved organic carbon that most animals cannot. And then they not only consume it, but then they fuel the productivity of coral reefs by shedding particulate organic carbon somehow. From my data from my dissertation, I didn't think that was the case. My findings kind of confirmed that, so we refuted this hypothesis. And then I spent a couple years as a sophomore professor at the University of North Carolina, further going down the hole of how sponges influence nutrient and carbon cycling on coral reefs. And then COVID hit. This position opened up at Old Woman Creek. And I went from the marine realm to wetland, aquatic freshwater sciences. And whereas, I study what goes in and out of sponges, I now study what goes in and out of wetlands and estuaries, and how all the different biogeochemical and ecological processes affect nutrient and carbon cycling. So real quick for my talk, I'm going to just talk real bit about Old Woman Creek, who we are. And then we do a lot of water quality and quantity monitoring. So I'm going to talk about these 4 programs. And if I have time, I'm going to give some examples of how we use that monitoring information to inform research. So a bit about the National Estuarine Research Reserve System. Is currently a system of 30 reserves around the country, most along the salty coasts. We're among 2 in the Great Lakes, the other being Lake Superior. There's another proposed, will be coming online in the Bay of Green Bay, but it's a federal-state partnership. Every reserve's federal partner is NOAA, the National Oceanic Atmospheric Administration, and then the state partner is typically a DNR or an academic institution. Our state partner is Ohio Department of Natural Resources. We fall under the Division of Water Resources. And basically our work is broken down into 5 sectors. So research, monitoring, stewardship, education, and coastal training. And as the research coordinator, my job basically boils down to conducting and coordinating research, applied research that we can apply to education and to address management needs for Lake Erie and coastal ecosystems. A little bit about what makes Old Woman Creek unique and why it was selected as a reserve. We have this natural barrier beach. It's one of the few remaining on Lake Erie and within the Great Lakes. Most of Lake Erie, the shoreline is hardened either by, rocks, concrete, metal. But this beach naturally opens and closes. So it's closed here. If we have a large storm event, water levels will rise. Often the water level of Old Woman Creek is higher than that of Lake Erie. When that happens, then you get this barrier beach break and water levels can drop several meters in a matter of a day. So this is some depth data over, I think, a year. And you can see all this variation. You know, these spikes represent a large storm event. And then after the storm, the barrier beach opens and you get this sudden drop of several meters in water. So getting into some of our water quality monitoring, what we're primarily funded to do lands under what we call the System-Wide Monitoring Program, or SWAMP. And this primarily consists of monitoring water quality and weather. And we've been formally doing this since 1996. Water quality. So we're located up here, Huron, Ohio. We're the southernmost portion of the Great Lakes. The green is our actual boundaries. So all these blue dots, 1, 2, 3, 8, 5, and 6, those are long-term water quality monitoring stations. And then we also monitor weather and precipitation. We have 2 weather stations, one up here and one down in the southern portion of the watershed, and that's because precipitation patterns vary a lot between the coast and down south. Recently, we've begun to synthesize some of this long-term data across the reserve system. This is some results from a recent synthesis, and this is a trend in oxygen, water temperature, and chlorophyll A as a proxy of phytoplankton over time from 1996 to present. Walk you through these. So this is dissolved oxygen. Oranger colors indicate that oxygen has decreased over time. Oxygen is really important for aquatic wildlife such as fish. For the majority of reserves, we've found that widespread hypoxia or anoxia over time. If the circle, so each circle is a different long-term monitoring station. If it's filled, then it's a statistically significant trend. If it's open, then it's not significant. So for us, we have observed significant decreases in oxygen over time. If you look at temperature, again, the majority of water quality stations have observed increases in water temperature. For us, these increases have not been significant yet. And then for chlorophyll A, due to loading of nutrients or eutrophication into the estuaries. Again, the greener colors indicate an increase in chlorophyll over time. So both us and the majority of reserves and water quality stations have observed a greater amount of chlorophyll A over time. So indication of increased algal blooms. Another water quality program that we're heavily involved in is H2 Ohio. So if you're not familiar with it, it's Governor DeWine's effort to mitigate harmful algal blooms in the western portion of Lake Erie by reducing the flow of nutrients to the lake. ODNR's role in H2 Ohio is constructing wetlands. I don't construct wetlands, but our role, along with a group of academic researchers called the Wetland Monitoring Program, we monitor a handful of these wetlands to see how well they're performing, to try to inform the construction of wetlands. This is a map of all the, it's probably not an updated map, but we're up to over 400 wetland projects across Ohio. Just to give you guys a flavor of some of the wetlands that I monitor. So this is Duck Creek out in Toledo. This is a wetland, well, not yet to be a wetland, but this is using beneficial reuse of dredge sediment from Sandusky Bay. This is Cedar Point Causeway. Cedar Point is up here. So if you go out to Cedar Point, look left. This will be a wetland in a couple of years. This is out at Pickerel Creek Wildlife Area. This is Pickerel Creek floodplain. Adjacent to is Raccoon Creek wetland. Then two other that we're just starting. They're still under construction, but Pickerel Creek East and West nature-based shoreline. So the idea is so this is Sandusky Bay, this is shoreline, and this is going to be a wetland beyond this shoreline feature. Duck Creek out in Toledo, this is a wetland that we've monitored for the longest period of time. So I figured I'd show you data from that. What you're seeing here is difference in total phosphorus, dissolved reactive phosphorus, and total nitrogen. And this is the difference between water flowing in and flowing out. So this is a flow-through system. We can compare nutrients coming into the wetland, nutrients coming out, and the difference is theoretically what the wetland is retaining. If it's above zero, then the wetland is doing a good job of retaining those nutrients. If it's below zero, then it's a potential source of nutrients. So we see that for total phosphorus, dissolved reactive phosphorus, the wetland seems to be doing pretty well at capturing, holding on to those nutrients. Nitrogen, a little bit more variable, potentially a source in the first year, but potentially becoming better over time as wetland vegetation take hold. There is an annual report out. I won't read all of this, but we do see among the wetlands that have been monitored to date, there is consistent nitrogen and phosphorus reduction by these natural filters on the landscape. And the 2025 report should be out here in a couple months. Another long-term and monitoring program that we conduct is what we call our nutrient retention and loading program. So this is our longest term data set, extends back to 1980 when we were founded. And just as Duck Creek, it's basically a flow through wetland. Old Woman Creek is very similar. So we have a well-defined inlet, well-defined outlet at Lake Erie. Water flows through the inlet, goes through Old Woman Creek, and exits into Lake Erie. So at both the inlet and outlet, we have auto samplers, and they're sampling water every 8 hours. And then every week we go out, we take those bottles, we bring them back to the lab, and we can quantify nutrients in those samples. Some examples of how we've used that data. So some colleagues from University of Toledo were able to use it to basically model how wetlands work. So what you're looking at here is this is phosphorus concentration exiting the wetland, phosphorus coming into the wetland. So as phosphorus coming into the wetland increases, the wetland's pretty much retaining those nutrients. But then you reach this threshold beyond which, if you keep increasing the inflow of nutrients, the wetland performs less well at retaining them. So they do really well until you reach this threshold. Another program that we use, so we're pretty limited by capacity, so we use volunteers to monitor not only Old Woman Creek, but also neighboring tributaries. It's for a couple purposes. First, to better understand our streams, so to obtain more data, but also it's an outreach and educational tool. So if you're familiar with the Sandusky area, we monitor the Mills, Pipe, and then further east, Old Woman and Chapel Creeks. Every one of these dots is a sample point where volunteers go out and collect water. We have approximately 25 volunteers a given year, but they go out, we give them kits. They grab a water sample for us that they bring back to the lab. They also have handheld YSI meters. So they're measuring things like dissolved oxygen, turbidity, water temperature. And then we go out with Erie Soil Water Conservation District, and we measure macroinvertebrates, which is a biotic indicator of how of stream health. And then we get all this data at the end of the year. Using some methods from the University of Maryland, we're able to put a letter grade on water quality for each of these streams. And we've been doing this. I'll take questions at the end, if that's okay. We've been doing this, I think, since 2012. So D plus C. The only reason Old Woman Creek is doing better than the rest is because we have a wetland at the mouth of Old Woman Creek that's doing really well at retaining nutrients. These are both available at our visitor center, but we also mail them to everybody living adjacent to these streams. And then a broader effort, there's a lot of these volunteer monitoring groups around Lake Erie. So I think 5 years ago, the Cleveland Water Alliance basically coordinated all of these groups. And this is kind of an outdated slide. There's actually more partners now. But what we've done is standardize our monitoring protocol so everybody's collecting data in the same way. And actually, just this year, the Ohio EPA is now accepting our data. So it's pretty credible. And at the end of the year, every 2 years, we're able to use all our data to pretty much report on the health of the water flowing into Lake Erie. All right, a couple examples of how we use our monitoring data to inform research. So this is one from Old Woman Creek. Some researchers from Ohio State University, they're taking tree cores from large trees adjacent to the Old Woman Creek, and they're able to measure the chemistry of each of the rings in those tree cores. And of course, each ring is also an indication of how old those rings are. So they're trying to, by pairing the tree ring chemistry and the water quality data that we've collected, trying to look back and see if they can reconstruct water quantity and quality using that data. And if they're successful in using this approach, they might be able to apply it to other systems such as the Maumee River, which is really the main source of nutrients to the Western Lake Erie Basin. We have what's called a Meriflex tower within our wetland. So it's measuring methane. These towers are located throughout the globe. Ours was the first in the wetland system. This is a graph of methane flux. We're really interested in methane because it's a potent greenhouse gas. It's like 25 times more potent than carbon dioxide. Mean annual air temperature. So if you compare wetlands at a similar latitude or temperature to us, and this is a log scale, Old Woman Creek emits order of magnitude more methane than similar wetlands. So the big question is why? So that's prompted us to now monitor carbon in the water. We're thinking, so this is measuring vertical flux of carbon. It also measures carbon dioxide. But we're thinking that it might be the lateral carbon in the water that's being contributed to the high methane emissions. And then finally, our water and meteorological stations, we use some of the highest quality sensors that are out there. We've begun using our monitoring stations as a test bed for novel and cheaper environmental sensors. So I mentioned the H2O monitoring work. That's a lot of wetlands to monitor, and researchers can't afford to put, $15,000 sensors at every wetland. So we're testing cheaper alternatives. So these are some cheaper alternative weather stations. These are water level sensors. This is a novel type of buoy. A drone that's able to sample water from distant or difficult to access environments. And then really cool, this winter we tested So we have to pull our sondes or water quality probes whenever they're threatened by ice, because the ice can shear the cables. So we don't really have a good handle on what's going on under the ice. But this device, you could put a sonde on it and it uses electromagnetic waves to transmit that data, and it can transmit through ice. So that's the first time we've been able to do that this winter. With that, I appreciate the opportunity to talk to you guys. We'll share this. This is a QR code for, I think, the next seminar, and I'd be happy to take a few questions. Yeah.
The QR code, you'll see it every week. It's the same code. It's for your, it's a survey for how well you like this format. So not necessarily Steve's presentation, but I mean, you can make comments on Steve's presentation, but
I won't see him.
the whole thing. You'll get the same question next, So you scan it, fill it out. It helps us plan future lectures. Any questions? He's got to get to the ferry soon, so questions.
You have questions? Yeah.
So how does, like, can you tell me a bit more about the grading system? Like, what would, like, essentially, what would be desired of a wetland, of a river, in order for it to get an A?
Well, so a couple of things that we measure. So sedimentation, a lot of these rivers. So we're seeing increase in the intensity and frequency of storm events. So just the other day, we got an inch of rain at the reserve in a matter of hours. And when you get those large precipitation events, that's when all the sediment and nutrients from the landscape runs into the tributaries. So sediment, that's not good for things like fish or macroinvertebrates. Nutrients is another one. We were seeing a lot of eutrophication or increase in nutrients over time. Decreases in oxygen, that is important for wildlife, aquatic invertebrates. Increases in temperature are not good.
It'd be clear water.
Yeah, clear, cold, nutrient, low water.
Your time at OWC, are there any projects you're like very involved in or ones that you've gotten off the ground that you haven't mentioned yet?
The carbon one's the big thing. So I kind of briefly talked about my sponge work and feeding work, but there hasn't been a lot of carbon work when it comes to water quality in the area. So that was the first thing that I kind of started. We got a new analyzer, started measuring carbon in water. That's probably the biggie. We're also looking at nutrient limitations. So phosphorus, or I'm sorry, I'm going to say this wrong. Yeah, phosphorus is typically the limiting nutrient, or thought to be the limiting nutrient in freshwater systems, whereas it's nitrogen in marine systems. That typically holds for Old Woman Creek, but when we've analyzed some of our historical data, it actually looks at like we might be nitrogen limited for most of the growing season, which is kind of interesting. So we've been doing some incubation experiments and dosing different concentrations of nitrogen versus phosphorus, kind of tease that apart. Yeah.
Can you briefly explain the AmeriFlux tower does?
Yeah. If you Google AmeriFlux, these are funded by the Department of Energy. And they're measuring methane, carbon dioxide. I think some might do nitrous oxide, which is another greenhouse gas. But it's a way to monitor these type of emissions over the globe. And this data can be put into like climate models to predict how the climate may change. All wetlands naturally emit methane. I didn't mention that. But the big question is, why does Old Woman Creek emit a lot more than other wetlands? We think that it might be because we're in an agriculturally dominated system, and we might have just a lot of carbon as well as nitrogen and phosphorus coming into the system.
And just kind of be here. And you ever come across like conflicts with all the different down one? When they probably want sampling done in certain states that they want, is that an issue?
So for the volunteer water quality program, before Cleveland Water Alliance kind of brought all of us together, there were a lot of groups monitoring different things or in different ways, different protocols. But over the course of just a couple of years, they were able to work together to kind of standardize things. Some groups monitor additional things, but at least for a select number of parameters, we're able to monitor in a standardized way.
Yeah, I remember being on those calls with Cleveland Water Alliance. Because I have a volunteer program with a charter captain. And like I did see it through the end, but I'm glad that they all got together. They were like, this is the way we always done it. I'm not changing. Yeah. And that makes all our data better. Yeah, it probably does, but I'm glad they got it.
And it's they initially had funding, so they were able to fund the different groups to come together.
Yeah.
But the interesting thing is for the last couple of years, we haven't had funding and everybody's still involved. There have been some groups that have dropped off, but we now have more groups than we've had before. And now that the EPA is accepting their data, there's credibility in the data with more people doing the same thing the same way.
Yeah.
Was there a process for the data to get accepted by the EPA, or did they just say like, hey, we're gonna pick this up?
No, so I think they looked over our SOP, which is pretty thick and detailed. We have like calibration logs, so everybody's calibrating and documenting calibration of their sensors that they use. We have people that have EPA certification to do water quality tests. Not everybody is certified, but there's a number of people within the group that have that like level 3 certification or whatever it is. So there are a number of steps that you have to take in criteria to meet.
What level are they certified? 1, 2, or 3?
I want to say 3, but only a couple. But we didn't need everybody to have that. Not everybody's. So we don't do nutrients. Some people do, but we're standardized for basically everything the YSI measures.
It's a pretty lengthy process to get that credible data. Let's do one more.
I have a question.
Yeah.
When you're designing a wetland or just like a buffer zone, how, like, generally speaking, I know every site's different, but how long do you expect for those to get the results that you plan for? Like, how long does everything take to really like set in, start getting data that's valuable for the standardization?
So every wetland is unique, and if you go to 2 different H2 Ohio wetlands, they're going to be different. But in terms of like design, you basically want to slow down water so that you allow sediment to settle and give plants an opportunity to take up the nutrients before it exits the wetland. You want a connection to nutrient-rich water flowing into the wetland. You don't want it to be isolated. But in terms of like development, Duck Creek, I think this data, I mean, we'll see. We're going to continue to monitor, but it seems like it's doing better over time. And I think it's because the vegetation community is becoming more established. That's just my hypothesis, but this is a really unique wetland because it's in the Port of Toledo, and I have to wear a hard hat and a vest to access this. This is a Cleveland Cliff steel. Well, they don't produce steel, but they produce whatever you need to produce steel, the pellets. So a very industrialized area. Duck Creek flows into the Maumee River. So it's a small project, but it's kind of cool to see over time. Yeah.
Do you find that developing wetlands near brownfields helps with the remediation or the seeping of toxic substances into the, I mean, the data show?
I don't measure that, so I'm not aware of it. But this was a former brownfield.
All right, let's thank Dr. Steve McMurray one more time. And hope to see you back online and then yeah. So good luck with your finals if you're leaving. And see you next week. Stop.